These are some questions from the student file. So let's try to talk about some of them. How does the structure of a molecule tie in with a function? That's actually kind of the whole point of the next one of the next FRQs we'll have as soon as we have time to have FRQs, which is the whole idea that of the last lecture that we have. There's a lot of examples that I've talked about in the EKs. Talk about. If you look at the EKs, it's pretty clear what they want you guys to know. All right. So review the EKs and make sure that if you lost, review the last lecture. It's recorded, right? But basically, in the last lecture, I talked about what difference occurs to function when you have a different type of bond in a carbohydrate that allows you to make a brick wall of sugar instead of a straight line of sugar, right? So then you get a structural function instead of an energy storage function. The glycogen branches out, allowing more surface area for digestion and quicker release of the energy. Meanwhile, starch is a long molecule. Well, plants don't need to move around, right? So we're talking about these differences in function. DNA versus RNA, they're different because of different function. I talked about how RNA has uracil and ribose because it makes it more stable with a single helix. DNA has to be a double helix because it gives you that spell check mechanism of having the second base to make sure that, how would you know there's a mutation, right? If there was not a second strand to tell you that the other side is off, right? Based on base pairing rules. So because it's a molecule that's supposed to be storing information across generations, it makes sense to have that spell check built into it, right? We talked about the fact that ATP is a molecule that doubles for the energy and for the glucoside trisphosphates are the molecules that double, they carry energy in the cell, but they also are the building blocks of the genetic information. So again, the structure begets the function, right? So you have the ability of the molecules like RNA to self-replicate because the building blocks themselves have the energy that's necessary to make the replication happen. So that makes it's interesting. We talked about lipids, right? So you have waxes are box-like with rings, structural support, right? Triglycerides, long chains of fatty acids, storing energy, insulation, things like that. Fatty acid chains, kinked versus not kinked. One is more liquid-like, one is more solid-like. It would change which one you would have in a cell membrane, which we'll talk about today, changes depending on what your need is, right? So if you have a need of a more liquid-like membrane, then you probably want to have more kinks, right? More unsaturated fat, which is more the type of fat that usually plants do it. But animals do that too. When they need a more fluid membrane, like say, for example, you live in a cold place, right? So you have more fluid phospholipids. That's also another modification that talks about functional change. You put a phospholipid, a polar group on the top of the lipid, and all of a sudden you have a molecule that doubles as both hydrophobic and hydrophilic, right? I'm not going to repeat the entire last lecture, but if you, basically that is what the EK is about. It's not even so much the chart that we talked about. I told you, fill out the chart. You're going to want it. You're going to need it. But the last column, which is what I said I was going to do for you guys, and this is what I did last lecture, it's basically how the structure ties in with function. One thing that I did very much hurried last class was the idea of directionality, though. And that matters because, you know, a protein will always start with the amino group in the front and always end with the carboxyl group at the end because that's how the backbone of the amino acids are built like. That means there is a beginning and an end for the protein. There's a start and an end. Likewise, the DNA code can only be read in a certain direction to build a protein in a certain direction. If you read in the wrong direction, you get the wrong message. And so the molecules have to be able to identify the correct direction for these things to happen. So directionality sometimes matters too. And we'll talk more about that when we talk about DNA replication and later in the year. Now, what affects the directionality of the polymer? Like I said, if you're a protein, all right, you're going to have to want to start with the amino group and want to end with the carboxyl group. So that tells you, whatever part of the body is looking at that protein. For example, today we're going to talk about, if we have time, we'll talk about the RAF-ER. And the RAF-ER has these little ribosomes in it. But they're actually not living there. There are free ribosomes that go and become attached to the RAF-ER because they're making proteins that are going to be exported. How do they know? They know because the very first amino acids of a sequence of the protein that comes out of the ribosome, it's basically can tell the cell where that protein is destined to go. So there's this special type of RNA called SRP RNA, which is single recognition particle, and then they actually detect those initial amino acid sequences.
Good morning, Matter Lakes teachers. Please pardon this interruption. Teachers, please do not forget today's meeting at 1:15 in the library. I'm sorry, not the library, in room 520 at 1:15. Even if you did not sign up for the PD, you must attend the meeting. Have a great early release day.
Okay, so directionality. So that signal recognition particle will tell the rest it's an RNA molecule. So you have the ribosome, and then let's say there's a protein coming out of it because it's being produced already. We're going to talk about protein synthesis later in the year. But the very beginning of that protein, the very few first amino acids of that chain, that sequence of amino acids serves as a message to this SRP molecule, right? Which is a combination actually of RNA and protein, right? So it will go there, it will attach itself. If you have the right sequence, right, it will attach itself to it and drag it to drag you and the ribosome to the rough ER. Because that signal told the cell you're going to make a protein to go to a place that's outside. But that matters then. It matters that you know where the start of protein is, right? So that's all there has to do with that part of the EK. It's basically the idea that direction matters. That it matters to say what's the start and what's the end. Likewise, the ribosome, when it's reading the RNA code, it's got to read on a certain direction so that it builds a protein in a certain direction. So there's mechanisms so the ribosome can recognize where the start is, like the start codon, all right, or the GCAP, which are the parts of the ribosome. We'll learn about this later in the year. But they know where the beginning of the RNA molecule is, right? So they can read from the beginning and build a protein correctly. Likewise, the RNA code, the DNA code, has a direction where you're going to start reading from and a starting place from where you start copying the code. So all that the EK is talking about really is that directionality can matter as well. And it certainly matters in proteins, RNA, and DNA. All right? And that's what the EK goes into. But basically, review what the EK says. Make sure you know the examples. I talked about them in class. Review the video if you lost, and that's kind of how it goes. Remember that this, as any other lecture, this is not all-inclusive. You study what I talk about here, you can probably do all right in the FRQs. But for the test, you need to study the book. You need to read it. If you don't read it, you're going to be caught. In the X marked sections, what are some examples of the specific molecules that are beyond the scope of the course of the DAP? What they mean is that, I know what you're talking about here. You don't need to know the actual structure of the proteins, the actual structure of enzymes. I'm saying you need to know how hemoglobin looks like. You don't need to know, are you with me what I'm saying? It is advantageous for you to recognize what a phospholipid looks like, what a fatty acid chain with a kink looks like, what a fatty acid chain without a kink looks like, what an amino acid looks like. Oh, that's a protein. So that's when I put in the chart. You need to be able to recognize the shape of the monomers and the polymers. But they don't want you to know, like, estrogen. What's the actual molecular structure of estrogen? Are you with me, I'm saying? They don't want that. They want you to know in general what waxes look like, the rings and stuff, or the things like that. Anyways, these, we barely will, what is this? Cytology quiz, chapter 6 question. What did you get it from?
I got that like on the quiz.
Oh, the quiz?
Yeah.
For your actual quiz.
Yeah.
So questions like that, I can answer to you in like on a one-on-one basis or at the end of class if there's time, okay? Because I have time left. Go ahead.
We have to start the uniform.
Uniform check. Stand up, please. Now, you can keep paying attention. Say, eyes on the target. Now, for lab number 3, part B, for gene name, do we put the actual name of the gene? Yes, gene 1, gene 2, gene 3, whatever the gene name of the file actually was. All right? It's a bit late for that, though, because that was due.
It's due next week.
Oh, it's next week. That's right. All right, so you know that. By the way, part B is due next week, so stay on top of that. And also this week we have lab 4. Okay? We have pre-lab 4 now. We are supposed to do part A of the lab in class next class. But that's okay. All right? But I don't know for sure if we're going to finish what we have to talk about today. Most likely we aren't. We only have 45 minutes to cover 2 topics. So the chances of it is very small. If we don't finish it, it's possible that I might have to push things forward. I'll let you guys know on Sally, all right, how I feel about it. But Either way, the pre-lab is due that day in class. What I'm trying to say is that we may not get to the lab next class. Are you with me what I'm trying to say? Okay. More than likely we aren't. I'm just saying like what's probably going to happen is I'm going to try to crunch this lecture and the next the lecture because we're behind because of the day we skipped. We're going to try to crunch the 3 lectures of these 3 days in 2 days, and then maybe do an FRQ next time, and then the lab will be the next time. So don't worry about it. I'll clarify everything on Saturday. Now, for measuring the plant's biomass, how do we find the dry mass of the plants? This is a question about the pre-lab for lab 10. If you read the actual investigative lab manual, it talks about what you're supposed to do. So you're supposed to dry it. So you put it in the stove, but it's inside. Okay. So we actually measure the mass on a little aluminum foil, right? So put an aluminum foil, put the plants on top of it. You measure the wet mass, and then you put it in the stove for 24 hours. You come back and measure it again. All right, and that's how you get the dry mass, because the water will all evaporate after it sits on the stove for a while. It's a very low temperature stove. It's not going to bake it, but it's going to make the water evaporate. It's going to dry it. It's going to desiccate it. How do we differentiate the mass gain from when we start growing them? Well, when we start growing them, how big were they? They're seeds, right? If you put all 300 seeds that come in a packet in a scale, you probably won't get a gram. Are you with me? So obviously you basically start from 0 grams and you go to whatever mass you go. And we'll talk more about that when we do the actual first collection of the lab. I didn't even see if they sprouted already. Let's see. Okay. So today's early release day. I don't know if anybody can do it. If you can do it, great. If you can't do it, we can do it tomorrow after school. Except I won't be here tomorrow, so you guys have to do it on your own. But all you can do Wednesday after school. But as soon as possible, we gotta get the competitors out of their same thing. Because remember, if you came here on thing, we talked about this. We planted. We didn't have enough seeds to plant all the quads, so there's extra quads left in there. They're in order. What you're gonna do is get the quad out and transplant one of the plants that's growing, because you're not supposed to have one. How many per thing? 1. So we're gonna do is transfer one of them carefully with the root and everything else, and plant it on an empty quad, right? So we can use every single plant we have. They're already sprouted, all of them. So then we can do that immediately, So if you guys can do it today after school, great. If you can't do it today after school, you can do it tomorrow after school. You can do it during lunch. You just come by and help. We're also going to reorganize the groups into new groups, and I'm going to put it on this on the tracking sheet. There's going to be a new column. If you go scroll all the way to the right, all the way to the right, tracking sheet, there'll be a new column that says planting group, and you will see A, B, C, D, and whatever. And then tomorrow we'll start the actual planting care groups. All right. So, but basically the group that's going to be tomorrow is like if you're a multiple of 4, a number multiple of 4, you're probably going to be in the group that comes tomorrow. Most like, how many people are in this room? 20. What's the last number? 30. No. So if you're a multiple of 5, we're in this picture, guys. The group is 6 this time, because it gives you more leeway to knock. All right. So is it? Are you 30? Not 30. There's people dropped out. It's like 28 now. Anyways, I'll figure that out. I'll let you check the tracking sheet today. On the there's going to be a column towards the right that will tell you your planting group. Tomorrow we'll start with group A. If you're group A, check out, come by, water them, take care of them. We have to take better care of them because in the past they do so hot. If the wick is not wet, it's a bad sign. The plants are going to dry out, right? Soak the hell out of that cloth. It should be picking up the water, but if it's not, soak it. Make sure that it's staying wet. All right, let's get to what actually they have to talk about. When I finish, you ask questions. If you have questions, write it down. Okay? If you have questions today, sell it, write it down. All right, because they have a lot to cover, and I'm going to make sure I finish it. All right. So the first question, the first case is about the cell membrane as a selectively permeable membrane. Now, what that means to be selectively permeable? What does that mean, Paul?
It lets in what it wants to. And
Exactly. It's true. It lets in what it wants to. So let's talk about some of the things. Make sure to get leaders to participate. Tell me something, Samantha. One thing that the membrane allows through. No, just allows through, like it's not even there. Passes through it freely. Water. Incorrect, actually. Water does pass through freely, but because there's something in the membrane called aquaporins, they're like little channels. So actually, water passes through to facilitate diffusion. If it wasn't because of that, it wouldn't pass through freely. Now, you're not entirely wrong because some water passes through. It's small enough that some water molecules will go through. So yeah, you can say water, you're going to get some water going through with no problem. But water does need aquaporins to go through. Now remember, aquaporins are a facilitated diffusion. All right? We'll talk more about that in a second. Still, Samantha, try another one. Something that will go through. No problem at all. Okay, so again, this is not something you don't know on the first day of something. You've got to know this, right? When you don't know, when I put you on the spot and you don't know, you got to worry about that. Because the test is much harder than these basic things. So you got to make sure, and that's explicitly on the EK that I'm looking at. And last night you read it. You went over it, right? So you should not be in that position. I'm not talking about you, I'm talking about everybody. Okay? Now, so let's back it up first of all. So we talked about what selectively permeable means, right? Now, let's back it up a little bit more and talk about what the function of the membrane as a whole is. So what is the function of the membrane, Christopher?
To create a homeostatic environment?
Yes, to help create a homeostatic environment. All right, to basically divide the inside from the outside and control what comes in and comes out. It will be like a border patrol, right? Customs, all right? So that's basically, it selects, it creates, and that's the very first thing against. The cell membranes separate the internal environment of the cell from the external environment. So it's literally a barrier. Now, are cell walls going to do the same thing? The answer is no. Cell walls are not going to do the same thing. So cell walls are not protective barriers. Okay? Cell walls are support for most cases. Okay? So cell walls are junctions. Now, it is true that in bacteria, for example, the cell wall made of peptidoglycan provides an extra layer of protection that makes it harder for anybody trying to destroy the bacteria to destroy it. Some of our drugs actually don't work in some of the bacteria because of their membranes that some of the cell walls that some bacteria have. All right? So, like gram-positive, gram-negative bacteria, the difference is between, it's like how thick the cell wall layers is. In fact, with those of you who are, like medicine, stuff like that, when we talk about bacteria, we'll talk about that. There's some medication that does not work on some bacteria because of their cell walls. So it's not like it's not going to give you protection, right? It's not like the plant cells, it's actually more for, but it is more for support. And in plants, right? In plants, that cell wall has a secondary function also, which is also for homeostatic control. Because the plant cell likes to be in a state of getting a lot of water going inside, right? On a hypotonic state, so to speak, right? It likes to be salty inside, so that water gushes inside. So actually plant cells are constantly pumping active transport salt inside, even though there's already salt inside. Why? Because maintaining salt inside allows water to flow inside, which makes the plant turgid. The cell membrane will, this is one of the things we do in the lab, right? On the lab 4. If you already started the pre-lab, you know that's one of the things we do. We're going to put them in the microscope like Elodea plants, and we're going to see when we put salt on them, the difference when you put salt versus when they normally look like. So we'll take them out just from that water, we'll put them in the microscope, we'll see how they look like, and then we'll repeat it after putting salt on top of it. And you can see that the membranes, if you put a lot of salt on the outside, what happens to the shell? It shrivels up, right? However, although the cell shrivels up, the membrane shrivels up, the actual shell shape does not shrivel up because of the cell wall. And that means that since the bark of the tree is made of all these cells, the cell wall, imagine what would happen if a plant lost water. What would happen to the plant? It doesn't have bones, so what would happen to it? It would just shrivel up. And it does ultimately wilt, right? Because it loses the turgor, but it doesn't wilt at once, just because it's not in water. Are you with me? Or even if you put salt on the plant, it won't wilt at once, because the cell wall will maintain the shape of the cell. Right? Even if the membrane shrivels, the shape of the cells maintain. So it's more of a structural / shape function than a protective function. But that doesn't mean that parasites are not hindered because of the cell wall. So if you have a bacterial parasite trying to invade the plant, it is harder because the plant cell wall is there. So the bacterial parasite is going to have some cellulose digesting enzyme to be able to get through that. So all of these things are important. But anyways, back to the cell membrane, it's a protective barrier, right? But protective in the sense that it is selectively permeable, allowing certain things to go through, but certain things not to go through. So let's talk about some of these processes then. All right, we're still talking about things that need to get through. So, but before we do that, let's talk about structure that begets this function. So as you can see, what would you describe the structure of the cell membrane wall to be like? Natalie Rios, what was the structure, what does a name for it actually, of what the model for the cell membrane actually is? Do you remember what it is? But what is the structure like? What do you see in the picture?
A last one on top and on the
No, but the membrane itself, what is the structure of it? Like, what do you see when you look at it? What is it made of?
phospholipids and stuff.
A phospholipid bilayer. What else is in there? There's labels, so go for it. You know,
The substance, which is
what are these things?
canal.
They're canal things. What are they made of? Proteins. Now notice there's some proteins that go through the entire membrane. We're called the integral proteins. There's proteins that go back and forth into the membrane. They're called transmembrane proteins, and sometimes they're back and forth inside the membrane. This is another example of integral protein here. There's some proteins that sit on either the inside or the outside surface of the membrane, and they're called peripheral proteins. And then each of those proteins has a specific function. Now, why would you be only peripheral? Maybe you have a job associated with cell communication. For example, maybe there's a protein that's on the outside that sends the signal to the inside, but then that signal activates a molecule, which then activates this protein, which then activates another protein. And you're going to see this with cell communication. A lot of these peripheral proteins have communication roles. They're relayers of the signal. Okay? They also could be anchors for the cytoskeleton, or for the or for the extracellular matrix support that connects two different cells together. Right? So they could be anchors for other proteins, which will then attach them to each other. We're going to see a video about this, either this class or the next one, that talks about the inner life of a cell, and you're going to see these things that I'm talking about in a more clear way. But there's also proteins which have these carbohydrates, or proteins that have fats attached to it, right? Now we're going to talk about the Golgi apparatus later, and how it's and all the Raphael also, and how it's important for them to modify proteins. So how does this protein get a carb tail? It happened inside a Golgi apparatus. It happened inside the Raphael. They actually make modifications to the protein once completed. Sometimes they cleave it in half, throw the other one half out, use the other one. Sometimes they add pieces to it. Sometimes they add cofactors. Sometimes they add lipids. They take away stuff. All right? So proteins are not done when they come out of the ribosome. In a lot of eukaryotes, the proteins still undergo processing. And that's what happens in the Golgi apparatus and the Raphael. So that protein that's in the membrane most definitely went to the Golgi apparatus. That's why it's in the membrane, because it was packaged inside of a vesicle and then merged with the membrane. All right? Now, here's actually an interesting thing. When you merge to the membrane, we're talking about exocytosis now, right? So let's think about the membrane here. Let's think about if I want one of these blobs to merge with the membrane, right? Where do I want the protein to be if I want the protein to be on the outside? So let's think about that. When this thing merges with the membrane, remember this is also made of bilayers, right? When it gets over here, what's going to happen is that it's kind of like the opposite of endocytosis. And endocytosis is kind of like folds inwards, right? Now think about it. If I want to be, so like this is endocytosis, imagine, right? It's folding inwards. So if I fold inwards, this stuff here is going to end up on the inside of the membrane of the bilayer of the vesicle. Are you with me? Now, so that means if I want to go, so if I reverse the process and doing exocytosis, if I want to be on the outside, the protein has to be made what? On the inside of the vesicle. And if I want the protein to be here, it needs to be what? On the outside of the vesicle. Does that make sense? Now, if I want the protein to be exported, not be part of the membrane at all, but go into the intercellular space, then it has to be floating where? On the inside of the vesicle, so that when it merges, it will just be exerted. Are you with me what I'm saying? Now, all of this matters. So, for example, let's say I'm in a protein. I'm making a protein like insulin. I'm in a pancreas cell. I'm making insulin. I'm gonna throw this insulin into the blood, right? So, do I want, when I'm in the Golgi apparatus, does the Golgi apparatus put that on the inside membrane, the outside of the membrane, or the inside the membrane but not attached to it? Inside, not attached to it, right? So, because when it merges with the membrane, it will just go exocytosis and leave the cell, right? Now, if I want to make a protein that's for cell communication, a glycoprotein that serves to tell the body, I belong over here. That's most of these glycoproteins, which have these little tails, they do that. For example, your blood type is a glycoprotein. The A type, the B type, is a specific type of glycoprotein that tells your body, I belong here. I'm a red cell that belongs here. So these factors, right, which are there, serve as attachment sites between cells. They serve communication roles, all kinds of different things in the body. We're just actually beginning to learn about all that stuff. But it's like, recent science. But either way, if I want to put that on the outside, it's going to be on the inside of the membrane. But now let's think about this on the rough ER. So if you're looking at the rough ER, right, and then you have these little ribosome that just attach itself to the surface, and it's making the protein. Well, but this protein happens to be in the pancreas cell, and it happens to be the insulin. Well, what does the ribosome have to do? It's got to make sure the protein goes where? Inside the lumen of the rough ER. Why? Because inside the rough ER eventually be modified and it will stay on the inside. Now, if I'm making a protein to be on the outside of my cell membrane, the protein has to be done attached to the inside of the rough ER. Right? Because eventually this will travel like that inside the rough ER, attached with me, until it gets to the edge of the rough ER. And then it will butt out into a vesicle. But where will it be at that point? Inside, attached to the inside. Are you with me? So that means the rough ER will be like, at some point, it will travel to a certain point in the rough ER, and then eventually it will butt out of here, literally butt out, like a little bubble will form. But it will form like that, right? So they'll fold inwards and form a bubble, but it's still on the inside. Are you with me? So it will butt out of the rough ER, and it's still on the inside. And then it will go to the Golgi, it will still be on the inside. And it'll butt out of the Golgi, still on the inside. So everything, from the beginning, it's got to be secreted in the right place so that it goes out the right way. Are you with me? And that's why the signal recognition particle is so important, because it will tell the cell where and how to put this in the rough ER. So it's all interesting stuff. But anyways, notice that the membrane has a lot of parts. It has these proteins that have multiple functions. And I mean, I'm not a textbook just regurgitator. I'm trying to do analysis for you guys here too. But you should know the functions. We have structural proteins, we have communication proteins, we have carrier proteins that grab stuff from one side, grab it and take it to the other side. We have channel proteins which allow things to go through them. We have pumps. We have recognition proteins, a bunch of different proteins that do a bunch of different jobs, right, across this membrane. What else do we see that? She said proteins, the blob-looking things. I already talked about the glycogen parts, and then there's also lipid tails. So there's glycolipids. Glycoproteins and glycolipids, right? So the glycolipids is something like this that has the glycol part connected to a lipid. Either way, they're associated with cell recognition and communication. Are you with me? Now, these things then are like flags in the membrane that let the cell know who it belongs. There's something else that we didn't talk about. Cholesterol. These are molecules over here. What does cholesterol do? Let's see. Natalie Enriquez, do you know what cholesterol does in the membrane? You gotta know what cholesterol does in the membrane. Angelo?
It keeps the phospholipids close and not too...
Exactly. It's kind of like a glue to the phospholipids. Now remember what happens. If the phospholipids are getting close, is that more solid-like or more liquid-like? Solid-like. So what does the cholesterol do on the membrane? It make it more compacted and solid-like. So if I wanted to turn, this is important because it's changing the structure, changing the function, the last CK. If I wanted to turn this area right here, the membrane, into a sort of raft, because remember this thing, we're going to see a video of this in a second. This is more like a river. The whole thing is floating around. All right, so these proteins are not stuck there. They're swimming in the glycolipid layer. Are you with me? Probably next class. We're not going to get to it today. But this thing is swimming in there. So that means this thing is more like a river. So, but what if I wanted to create like a raft in this river? An area that kind of like make an island. Right? What would I have to do? And not only that, what else should I do? Because I want to make it more solid-like. Lots of what? Less bends. Saturated fat. So I would put a lot of saturated fat and cholesterol in that area, and it will become more solid-like. So that's how the membrane how membrane fluidity is controlled. Does that make sense? Now, so is cholesterol a bad thing? Of course not. It's a necessary thing. We need it, right? Now, so you talked about the membrane and it's all its parts, okay? This is called a fluid mosaic model. It's called fluid because it's all flowing around, and mosaic because it's built of many parts. But why is it, this is very important, what is most of the membrane made of? If you really look at it. Phospholipids, yes. But out of that, is it mostly phospho or mostly lipids? Look at it. I'll think about the volume of the membrane as a whole. You're looking at it. Is it mostly fat or mostly hydrophilic things? It's mostly hydrophobic things, right? Most of the volume of the membrane is made of the tails of the fat. So if you're getting through the membrane, right, you're going to have to deal more with hydrophilic stuff or more with hydrophobic stuff? Hydrophobic stuff. Now here's the thing about hydrophobic stuff. It doesn't just hate water. It hates anything that's like water. All right? So hydrophobic stuff loves hydrophobic stuff. So if you're hydrophobic, you get through with no problem. But if you're hydrophilic, you have to deal with the tails. And you're not going to get through. In fact, that's the whole point of it. Right? So what kinds of things get through with no problem? All right? Hydrocarbons. Like, very good. So hydrocarbons. All right. What else gets through no problem? Oxygen, because it's it's small and then nonpolar. Carbon dioxide, small and nonpolar. All right. Things like estrogen and progesterone and other fat-based hormones. Very important. This is why they allow you to communicate quickly throughout the entire body, because they are lipid-based molecules. So like lipid hormones, like steroid hormones like the estrogen and lipid hormones, they will be a good example of that. All of these things get through. Anything, some water, because it's small, gets through, but water is polar, so it's not going to get through very easily. One of the things that I think is important too, it is the surface tension of water pressing from both sides that actually helps maintain the shape of this. On top of that, if you look at the membrane at the bottom, it will have a network of cytoskeleton fibers providing a kind of like, at least in the eukaryotes, not necessarily in bacteria, but in the eukaryotes, it will have a network of cytoskeleton fibers that provides kind of like a scaffold for this membrane to kind of have its shape. So when I want to, say, for example, do phagocytosis, which is basically pushing my arms forward, right, to swallow something up, well, how does that happen? Basically, the cytoskeleton scaffold extends. They build more cytoskeleton, which pushes the membrane out. Are you with me? Just like, think about it this way. I'm at the cytoskeleton holding the membrane in place, right? And then when I want the membrane to go out, I just make this arm longer. Are you with me? And that pushes the membrane outwards. Does that make sense? So polymerization of microtubules is what's going to make that happen, because microtubules is the cytoskeleton component. All right? Very interesting. Conserved core structure among eukaryotes, cytoskeleton, which allows it to make pseudopod motion, something the bacteria can't do. All right? Conserved core process. All right? Motion through cytopods. So it's a structure that matches a process. All right. So anyways, this is the structure of the membrane. But it seems that the majority of the membrane is hydrophobic. Hydrophilic things will have trouble getting through. And then each of them will have to do something to get through. If it's a large thing, how does it get through? A very large thing, Kevin. Large, enormous thing, like a protein or a whole bacteria. No, it's too large. It's not going to get through in a chain. Either phagocytosis or pinocytosis. A large protein has to get through a vesicle, right? Now, phagocytosis is something enormous that's trying to get through. Phago means eating, all right? So the cell's actually extending its arms and swallowing the whole thing up and bringing it inside. Again, next class I'll show a video of this for you guys, all right? So if you haven't seen it, it's one of the videos about tutorials for cytology. There's lots of good videos and animations over there. Make sure you take a look at it, all right, if you're lost in this topic. So it swallows up. But that's called, the cell actually extends the cytoskeleton from the inside, pushes the membrane forward, and swallows the entire bacteria. And then that forms a huge vesicle, a food vacuole, which the lysosomes bind with to kind of try to digest the bacteria and kill it, right? Because the lysosome is full of digestive enzymes. Now, on the other hand, smaller things like maybe a protein or a larger set of amino acids or something that the cell wants to bring inside, but it's too large to go through a channel. Think of it as this way. Phagocytosis is like, I want to swallow a whole airplane. Pinocytosis is like, I'm going to bring some cars in. But if you want to bring car parts, then you can use the channel. You know what I'm saying? So that means it's a big thing still. You bring a whole car in. For that, you're going to need pinocytosis. Now, pinocytosis is the opposite of exocytosis, right? The membrane buds inwards and forms a little vesicle. Now, there's two types. There's one type that it just does that, and whatever's in there happens to go in. But there's a problem with that, because what if I want to get something specific in? Are you with me? Peanut butter means drinking. So it's like little sips. I think of it that way, right? So the cell is still budding inwards. And it's actually kind of cool to see, again, on the videos animation folder for cytology, there is actual videos of the cell doing pinocytosis, not like an animation. A video, a really close electron microscope video of the cell like budding inwards and making the pinocytosis happen. Right? So you can see how that works. But it's little gulps that it's taking out of the extracellular fluid, right? So it's taking some of the gulps outside, but it's not specific if you just do it that way. So then what a lot of them have is receptors. Glycoprotein receptors most likely. All right? And these receptors will sit there. When the molecule of interest attaches itself to the receptor, pinocytosis starts. That means it will not gulp inwards for any reason. It will gulp inwards when it needs to gulp inwards. Does that make sense? And it will have the thing that it wants. This is very... So for every one of these, think of a physiological example of it. So how about a physiological example of phagocytosis? Everybody knows this one. Natalie, Saborio, phagocytosis in the body. Come on, you can do this. Eating bacteria, but what's doing that though? What kind of cell is doing that? It's a phagocyte, yes, but what kind of cell is that? Oh, a white blood cell, right? It's actually called a mastocyte or a phagocyte, a neutrophil. All three of them do that kind of stuff. So they actually swallow up the invaders, right? Now, what about pinocytosis? A classic example of that will be, by the way, amoeba eating would be a good example of phagocytosis as well, right? When amoeba eats, it's basically a eukaryote that does that, right? Now, the amoeba eating will just swallow a bunch of things as well. Now, pinocytosis is a good example of this. Actually, nerve cells, your neurons, when they're receiving neurotransmitters. So when they're receiving neurotransmitters, they actually do it that way. The neurotransmitter attaches itself to the receptor, and then the budding in is what brings the neurotransmitter inside, which is going to either excite or de-excite the neuron to generate the action potential or cancel action potential that's in the process of being generated. So excitatory, so for example, if you get a bunch of serotonin being produced, right, makes you happy, makes you, dopamine. A lot of dopamine gets produced, makes you, happy and, alert, and maybe a little even anxious if it's too much of it, although that's probably a GABA. But anyways, too much dopamine. How does that get inside? How does that excitatory thing get inside? That way. Right? So there's dopamine receptors. So if I have schizophrenia, because I have too much dopamine being produced, right, I can create a blocker, a molecule that mimics this molecule, right, and enters there and sits on the thing and blocks the access to it. And then the dopamine cannot be received, and it will lower the dopamine expression. Or not the expression, dopamine reception. Are you with me? I'm saying there's one mechanism that I could use to target the disease, or I can actually suppress the expression of the gene that makes that protein in the first place, so I have less dopamine receptors. That's the other way you could do it. All right. So, or I can create something that attacks dopamine directly, or suppresses production of dopamine. But that's easier to do as inhibitor. And a lot of the depression drugs work that way, by inhibiting the receptor, or inhibiting the reuptake. A lot of these neurotransmitters, when they're not absorbed, they are reabsorbed by the emitting cell. So, like, say for example, I have the axon of one neuron here and the dendrites of the other, right? And they're waiting for, to receive that signal. When this guy sends a signal to the other one, some of the neurotransmitters which are not absorbed by the receiving cell are reabsorbed by the emitting cell. But they have drugs which are called selective reuptake inhibitors, which basically what they do is they block the membrane proteins on the emitting cell to prevent it from collecting it back. So that means those neurotransmitters get produced more, and then you don't have a norepinephrine deficiency in the synapse, which allows you to be less depressed, right? Because basically the neurotransmitters are not being taken back, so it's always at a high level to try to avoid the fact that you're naturally at low levels, and that's why you're depressed. So these kinds of drugs, see, understanding of biology, basic biology, actually helps with harder biology later, and that's why they call Introduction to Biology course. Because you go through this to survive the basics, so one day you can learn more complicated stuff. But anyways, the membrane structure has everything to do with this function, because if I have a full hydrophobic thing, it's going to have more trouble getting through because most of the membrane is hydrophobic. So now let's talk about smaller things, okay? By the way, exocytosis is just the same thing except launching it outwards, right? Now, those things are active transport because they do consume energy to produce, to make the vesicles or to change the, think about it, if I am doing pinocytosis, that means the destruction of cytoskeleton components to make this happen, and I'm going to have to activate that, I'm going to have to, there's energy expenditure there. There's energy expenditure to do exocytosis, I have to move the vesicles, which means I have to have a transport protein walking across the cytoskeleton, dragging that vesicle, merge with the membrane, produce, if I'm doing phagocytosis, I have to produce more cytoskeleton to push it forward. All of these things are kind of active transport, but not in the sense that you classically understand active transport, which is the idea of pumping, right? They're active in the sense they spend ATP, right? Now notice how that's different. You could have a lot of neurotransmitter out and none inside, and you still need active in the sense of building the buds to bring it inside. But technically, the neurotransmitters want to go inside. There's a natural tendency by diffusion for them to go inside, but they can't because of the selective permeability of the membrane. And it still kind of counts as active transport because you're doing that. What we mean in biology when we say active transport, though, is actually pumping against the gradient using a pump. But I want to make sure it's clear that it still spends energy to do exocytosis and pinocytosis and phagocytosis. Is that clear? You're spending energy because you're building those things. But what we mean by technically by active transport is when you're pumping. Now, pumping has to do with throwing something out when or throwing something in when there's already, we just talked about a plant cell trying to maintain a salty environment so the water, by diffusion, tends to go inside, right? So how do you do that? If there's already a lot of salt inside, what do you got to do? You got to keep pumping more salt inside. But the salt doesn't want to go inside because naturally there's a lot of air inside already. So if you have a gate that allows salts to go through, the first thing you got to do is close that gate, right? So that means there's gates in the membrane, right? But if you close that gate, right, change the conformation of the molecule, basically you have probably an allosteric activation of the membrane. So you're going to put a molecule there, a signal molecule, that will shut down, change the shape of the molecule, and close the gate. Now the salt is not going the way it wants to go anymore. Because remember, this is facilitated diffusion. When you have a channel or a gate or a carrier protein that allows, like aquaporin, the thing to go through the direction it wants to go through. The second thing you want to do is then pump. So let's say, for example, you have a lot of salt here inside the cell, and not so much salt on the outside, but you want more salt in there anyways. Like, for example, you're creating an active potential in a nervous cell. Here's another example. So I want to pump more of those positive charges outside to make sure the active potential happens. But the first thing you're going to do is close the channel that would allow them to naturally come back, right? Then I have to have a pump somewhere that constantly pumps that against the gradient. And how does that work? Basically a protein that attaches itself to the whatever cell is working on, and then uses ATP. When the ATP connects, it usually changes the shape, and then it kicks the thing outside. I have animations of this in the video animations folder if you want to watch how it looks like. Maybe next class I have time to show it to you guys. But basically, imagine like a triangle like this, and then you connect to it if you're the thing that needs to be sent out. Now, as soon as ATP connects to it, it changes shape, and that thing gets kicked outside, right? So it's a pump, so to speak. Sometimes it's more complex than that, like proton pumping in the chloroplast and mitochondria to make the proton gradient that allows photosynthesis and aspiration to create energy, all right? That is a lot more complicated. It actually involves the passing on of protons between several different molecules, so it's more like a carrier, so to speak. It's more like this. It's a series of oxidation-reduction reactions where the proton is being passed on from molecule to molecule and therefore across the membrane. So it'd be something like this guy gets a proton, passes to the next guy, who passes to the next guy, and then it passes outside. So it's less one protein changing shape and more several proteins passing the proton forward. Regardless of what it is, it spends energy to get that done, right? In photosynthesis, it's the sunlight that happens to provide the energy. In cell respiration, the energy comes from NAD and FDH that provide the energy, the electrons, to power the pumps. In your body, lots of things produce the pump. If you're talking about the neuron and its active potential, it's ATP that's producing the energy for the pump to occur against the gradient. Sometimes they actually recycle some energy. There's some co-transport system, so to speak. So there's a little protein here that's going to act like the pump, right? And then you have one that's a channel. So you have one thing that's a lot of it on the outside, and as it travels inside, that actual potential energy of something moving from high to low, like big water behind a dam to no water in here, that actual potential energy generates the energy that's actually used for the pump to pump something in the other direction. So there's an example of this in your intestines, the glucose co-transport. Basically, glucose has to go to the blood when you get digestion. And that's always true, even when you already have a lot of glucose in the blood, which is a problem. This is why we have insulin. Your body thinks about it, if I have a lot of glucose, great, I'll make some fat out of it, right? But I'm going to use it when I have it because I don't know when I'm going to have it again. It evolved that way because when you eat, you want to get as much sugar as you can from it so that you can, in case you don't eat again. You know what I'm trying to say? So the body constantly pumps sugar into the blood, even if there's already sugar in the blood. But sugar is a large molecule. It's not going to go in the blood naturally. And besides, usually the blood could be high in sugar already, right? But you want to keep putting sugar inside. So in the intestinal wall, that membrane will have pumps. And another thing. And then something going down the concentration gradient, I think it's a salt, powers the pumping of the sugar against the concentration gradient. So that's called co-transport. Right? The book talks about it. There's a chapter on, there's a section on it, so you can look at it in more detail. And there's an animation on the videos in Raptitoris folder as well. Now, so each thing gets to the membrane with some sort of help. So let's think about water. Facilitated diffusion. What does that mean? There's a gate, there's a channel, there's a carrier. In the case of water, it's aquaporin. Amino acids, glucose. There's a carrier protein or a channel. Something polar, like a salt, there's got to be a channel for it. Because these things do not go through the membrane. The membrane is selective against things which are polar, right? If I want to go against the concentration gradient, I'm using a pump, most likely. And then if I'm large, I'm using pinocytosis or even phagocytosis, right? And that basically is why everything that's over here. And I already talked about the other part, which is cell walls provide structural boundary, right? So in addition to actually, it is actually a permeable barrier, right? So like it's not selectively permeable like the cell membrane, but it does allow some protection. And plant cells have native cellulose, and then other species also have them. Like bacteria have peptidoglycans ones, and then fungus have ones made of chitin, which is another type of sugar, cousin of the cellulose. All right. So the next EK, 2.B.1, 4.A.2 actually, talks about the components of the internal components of the cell. Now I already alluded to this. I only have 5 minutes left. I already alluded this in the last class, and talked about the fact that why does a eukaryotic cell have internal components? For two reasons. Internal membranes increase the surface area to make up for the fact the cell is very large. Because a large cell has low surface area to volume ratio compared to a smaller cell. To make up for it, internal membranes increase the surface area. I told you, like, instead of doing a racetrack like this, if you do a racetrack like that, and you still get to the very end spot, you're still going to cover a lot more surface. And that's kind of the idea. Infolding provides an advantage for the cells by giving it more surface area for chemical reactions to take place. A lot of chemical reactions, which we'll learn about this year, happen in the membranes. And therefore, like for example, the production of energy in the mitochondrial chloroplast is a membrane thing. The majority of it is done to the ETC, which is a membrane thing. So that means we're going to, basically life has little hydroelectric dams, right? Or where it uses the proton, the fusion of things across the membrane, going down the concentration gradient to produce energy, right? And so membranes are important for life. A lot of the things that life does, it does in membranes. So increasing the surface area is one thing. The second thing is specialization. By having areas that do special jobs, you're going to decrease competition in chemistry speaking. An enzyme would have less trouble finding what it needs to work on, and we talked about this last class already, right? If it already it's working in a place where the things it's got to work on are there. It'll be, not having it would be the equivalent of having 6 different sports having in the same field at the same time. That doesn't make a lot of sense, right? And that's kind of why I talked about last class. What the EK actually asks you to talk about, if you look at it, all right, so we're not in biology honors anymore, so I'm not going to sit here and repeat basic definitions of what the things do. But if we focus on what the EK says, notice that ribosomes are universal structures. What does that mean? Conserve core structure. Everybody has it, right? Because you need it to make what? Proteins. You can't make proteins without it. So that's the first one that they mentioned over there. Because ribosomes are universal structures. So, and then, what is it, 4.A.2 that I'm looking for?
Yeah.
All right. And then they have two parts, RNA and protein. And those parts need to be assembled in sequential manners in order for their components to interact and do what they do. Now, what does the ribosome do? Translation, right? Production of proteins. So clearly you need to know what that is. And you need to know that every cell has it because building proteins are what cells do. They're cell factories. The ER, smooth and rough. It says you're supposed to know what they do. We talked about the rough already. It packages, modifies, cuts, adds, makes modifications to the things which are going to be delivered outside. And depending where you're going to deliver, you're going to make it in different places of the ER. Either in the lumen itself, or in the inner membrane, or the outer membrane, depending on where this thing is destined to go. Right? Now, ribosomes are not usually there. They attach there when the beginning of the protein tells the signal recognition particle that they need to be going to the right place. Now, the smooth ER is a lipid synthesizer. It also has a role in regulating toxicity in the cell. When things become toxic in the cytoplasm, what they do is they throw it inside the smooth ER. So instead of being the cytoplasm, it goes inside. So think of the smooth ER as a bit of a cleaning dump. Now, after that, it's inside the smooth ER, right? So then you just build a vesicle out of it and throw it outside the cell. Are you with me? I'm trying to say. So it's a way to export toxins from the cell. First you put it inside the smooth ER, and then you ship it out. Are you with me? So it means if you're a cell of the liver, what your smooth ER is going to be what? Just big, because that's what it does. But if you're a pancreatic cell, which exports proteins, what's big? The rough ER, maybe the Golgi. Does that make sense? If I'm a mitochondria, if I'm a sperm cell, what I have a lot of it? Mitochondria, because I'm swimming around like crazy. If I'm trying to say, each cell has a factory that's depending on what it does, right? If I'm like make sweat a lot, probably have huge rough ERs and Golgi. Does that make sense? All right. So it depends on what your job is. Now, the Golgi complex is a membrane cell structure. It looks like a flattened membrane sac, right? And it functions into packaging and delivering the things to where they go. So it's like the sorting unit that helps control all the deliveries to different parts of the cell. Not just the membrane, but different parts of the cell. Oh, you're a protein that's going to go over here. You belong to the mitochondria proteins. Go to the mitochondria over there. So it's like a central control agency to adopt that. The outer membrane is smooth, but the inner membrane is convoluted, full of folds. Why do you think the inner membrane is so convoluted, full of folds? To make more room for the job that it's doing, right? The mitochondria, a living endosymbiont that actually has primarily a role of producing energy in its membranes, but it also has its own DNA, right? The outer membrane is smooth, the inner membrane is full and convoluted. Why? More surface area for the actions to take place. The vacuoles. Vacuoles are basically specialized membranes to store things. Plants have a huge one and small ones. We only have small ones. But they're storage things. Special vacuoles that store enzymes to digest other things are called liposomes. Lysosomes, right? And they're going to be very common in cells that defend the body or cells that digest things, right? Chloroplasts, specialized living internal organelles in algae and plants, which, like the mitochondria, have its own protein factory, its own DNA, and it's basically specialized to capture sunlight to produce G3P, which is a building block that can be used to make a lot of things, including sugar, proteins, lipids, and nucleic acids, right? Now, next class, I'm going to start the class by showing you guys a video of the inner life of the cell. I'm going to show you a video of the processes of phagocytosis, exocytosis. I'm going to do a little animations at the beginning. After that, we're going to do the next HK, which is, and then after that, if you have time, we'll do an FRQ. And most likely, the lab won't be until Friday. Okay? Now, if your lab is still due next week, the next class, though, in case I change my mind. So make sure you do that. Now, notice that this is something that you need to know for honors. We don't have a lot of time to cover it. But if you have questions about anything in this EK or in the chapter of cytology where it talks about the functions of the organelles, make sure that you ask them next class when we have a chance to do that. I'm also going to show you some simple test questions that show you how do they ask questions about this, so that you can see what you need to know. All right? I'll see you guys next class. Yes.
My password for homework.
Again, this is fast and furious for people who already studied a lot.